One can imagine a surgical site being prepared. Bone, of course, is very vascular and the wound site fills with blood as the implant site is prepared. While wound lavage is necessary to remove debris created during the drilling process, blood will refill the site and start to clot. This is the recipient site into which the implant is placed. Two different phenomena can now occur. We can imagine that new bone would be formed on the old bone surface, called distanced osteogenesis, or we can imagine that bone would be formed on the surface of the implant itself, and this we call contact osteogenesis. The main components of each of these two phenomena are the parent bone itself, the implant surface, and the components of the resolving blood clot itself, including the new blood vessels that form as part of the healing process. Distance osteogenesis, we see a population of potentially osteogenic cells which arise on the old bone surface. They change their shape and become polarized osteoblasts and start secreting bone matrix. These cells, like all secretorily active cells, are dependent upon their vascular supply, so the presence of new vasculature, or angiogenesis, in this healing compartment is very important. But vessels can restructure themselves by capillary budding or occlusion, and therefore the capillary network can change in pattern as a result of the space occupied by the elaborated bone tissue. So you see that the vessels change in their position as the result of synthesis of bone matrix. However, as the bone matrix forms, some osteoblasts get buried in their own matrix as osteocytes. These cells maintain their cellular connections with the surface osteoblasts through the canaliculi in the bone matrix. Also, as some cells become buried as osteocytes, more osteogenic cells are recruited to the bone surface to take their place, and thus the integrity of the newly forming bone surface is maintained by continued synthesis. Of course, bone synthesis continues, although the vessels may move, and we end up with bone which approaches the implant surface but is separated from the implant surface by the osteoblasts themselves. As we have seen, these osteoblasts have their cell processes buried in the secreted bone matrix, and they form gap junctions with cell processes of buried osteocytes. Thus, osteoblasts are unable to migrate from the bone surface. The result, therefore, of distance osteogenesis is osteoblast death, since, as these cells continue to elaborate matrix, they cut themselves off from their own vital blood supply contained in the capillary network which itself has remodeled. The same basic biological principles apply to contact osteogenesis. But now we see the osteogenic cells appearing on the implant surface itself. How these appear on the implant surface will be the subject of the animation entitled osteoconduction. Here we are assuming that the cells have already arrived at the implant surface and are able to differentiate into osteoblasts, become secretorily active, and lay down new bone matrix directly on the implant surface. As they differentiate, they secrete the cement-line bone matrix, shown here in blue, directly on the implant surface. The cement line is well known in bone remodeling sites as the calcified, collagen-free matrix which separates old bone and new bone. The osteoblasts then start to produce the collagenous and mineralized extracellular matrix of bone which, as we have seen before, becomes populated by osteocytes. In this case, the final interface shows bone in direct contact with the implant. While it is most probable that both distance and contact osteogenesis occur in all peri-implant bone sites, it is clearly an advantage to have an implant surface which optimizes the amount of contact osteogenesis to stabilize the implant in bone matrix as rapidly as possible.